I'm delighted to be able to welcome you to this Oracles this evening. And I think this is in one of the most exciting areas of science and medicine today. My interest in this region really started way back in the late 60s, early 70s, when I became co-founding trustee of the then British Retinitis Pigmentosa Society, which was a patient organization designed to actually produce more research into the inherited retinal dystrophies. I was their chair of the medical advisory board for many years, and together with Alan Laties from Philadelphia, we were co-chairs of the International Prevention of Blindness group, particularly interested in genetics. I guess the story really started back in 1953 when a Nature paper by Watson and Crick highlighted the structure of DNA, and it was our first understanding of the double helix and the secret of life. Disappointingly, they left off Rosalind Franklin, who really generated the images that they were able to base their work. So roll forward just under 50 years, and we see another Nature paper, The Book of Man, the Human Genome. And I think this is fantastic in 50 years to go from a basic discovery to a discovery which is opening up the whole of human genetics, an understanding of control mechanisms in the human body. So if you look at where this work has led, it's led to, first of all, a complex group of scientists. In essence, we had initially the gene hunters. These were people who looked for defective genes and identified the chromosomes the defective genes were on. And then we had an even more industrious group, the so-called gene walkers, who walked up and down the chromosome and found the specific sites of error in the DNA. As it were, they were looking for spelling mistakes. Those people then collaborated with protein chemists who began an understanding of what goes wrong with the transformation of the defective gene into cell biochemistry. And again, we then have another coupling into the cell biologists who research into why cells die. And importantly, in this group, we see why a cell which is the only cell in which the gene is expressed, when sufficient numbers of them are killed, they kill the surrounding areas. So this is a very complex area. And just for those of you not familiar with the field, we now have very interesting protocols. In terms of autosomal recessive disease, we know that the parents are carriers, but don't necessarily manifest ocular problems. And so here, the concept of treatment is gene augmentation. We need to put more normal genetic material in order to compensate for the defective gene and hopefully overcome its action. And we see the early gene therapy trials which have used gene augmentation techniques. And of these, I guess the most spoken about is the treatment Luxturna, which is a treatment for Leber's and treating the gene RP65. Autosomal dominant diseases, where you have one fault and you have the problem, to my mind, in the early days, was going to be a real problem. However, it's really a gold mine in terms of gene therapy, because now we have two ways of coping with this. We can edit specific genes. We can and we can use an enzyme from bacteria, CRISPR-9, and we effectively silence the gene by cutting it out of the chromosome. Then we have true gene silencing techniques where we don't alter the chromosomal material, but we interfere with the messenger system between the gene and the production errors, production systems within the cell. So now a whole spectrum of potential for beginning to come to terms with genetic diseases. It is much more complex than this dumpy guide. So I'm absolutely delighted that we are joined here this evening by Professor Tara Moore. She is the Professor of Personalised Medicine at the University of Ulster. And to my mind, Tara is a true oracle. Given the original definition, oracle was a woman, a priestess, who told the truth because she spoke the word of God. Tara, tell us about how genetic diagnosis is undertaken and how you develop treatments.
Well, thank you, John. No pressure. Thank you, everyone, for joining me. As John has explained, we're all made up of DNA. In fact, 3 billion letters of DNA. And if you think of a genetic eye disease that is caused by just one single error, we call that a single nucleotide polymorphism. It's a mutation. There's a 1 in 3 billion chance of that spelling mistake in your DNA causing a genetic eye disease. The challenge, though, is for us is to find that needle in the haystack. And as you can see in the top left-hand side of this slide, I just give a few examples of some genes and some mutations that are associated with eye diseases. So far, 350 inherited eye diseases have been discovered that are caused by mutations in over 500 genes. So anything I described today, I'm confident is just the tip of the iceberg, and there's a lot of mistakes and a lot of diagnosis ahead of us. It's difficult for me in a small 15-minute talk to talk to you about the discovery of gene mutations and the classic linkage analysis that we conduct in gene sequencing. Large multi-generation families lend very, very strongly to the success of finding any genes. We do linkage analysis. We identify initially the part of the chromosome the gene is in. We look at recombination events. We come up with a list of genes. In that list of genes, we actually apply common sense and we think, well, are any of these genes associated with diseases that may be linked to this particular eye disease, or are they even expressed, say, if the disease is in the cornea, are they even expressed in the cornea? But it is very much looking for a library, a book in a library, a chapter in that book, then the correct page, the correct paragraph, the correct sentence, and then we have to find the mistake in the exact word. And that's what that discovery process looked like. Here you can see the TGF-beta-I. A large family was used to locate it to chromosome 5, eventually to an array of genes where TGF-beta-I was then found to be a causative mutation. Once we know that, when we have TGF-beta-I as a gene that mutations can happen in, we're able to go in and sequence exon by exon and find if it's an R124H or a particular type of mutation which is causing the phenotype or the disease symptoms shown here in this eye. And all of the eye specialists and cornea specialists amongst you today will be able to recognize each of those different corneal dystrophies by its presentation at the front of the eye. Avellino and others provide a rapid test for these types of mutations, and they use PCR. You'll all be so much more familiar now with PCR after COVID and its use during the pandemic, so I won't go into the detail of that. Within the one TGF-beta-I gene, there are over 70 mutations associated with lots of different corneal dystrophies, but 90% of them are caused by 5 very common mutations. There's a real-time PCR test, just like there was a COVID test that can detect that DNA mistake in anyone's blood sample or cheek sample. It's very simple sample prep. It very much reduces hands-on time. It's very straightforward interpretation. And the turnaround time for this is from obtaining the sample to be able to give a result is about 2 hours. So you can imagine that lends itself very well to a genetic diagnostic lab sitting within a laser surgical suite, whereby an individual is being pre-assessed for elective spectacle-free laser eye surgery, and they can be told whether or not they have this TGF-beta-I mutation, which would mean they would not be suitable for that surgery. But within that, we recognize there's about 65 other mutations in TGF-beta-I that we weren't able to detect and test for. So everyone then looks for a solution beyond it. And of course, it's next generation sequencing. So sequencing the genome, and everyone have heard of the Whole Genome Project and your whole genome being sequenced, so you can tell whether or not you're predisposed to certain diseases from Alzheimer's to cancer. So that's something that lends itself to sequencing for genetic mistakes for eye disease as well. Because of the increased usage of it over the last 20 years, there has been a great drop in the price, and it's now something that is affordable to be applied to eye disease. Specifically, I'll give one example of where we use targeted next generation resequencing. And that's where we're applying that to keratoconus. And what you do is you go into lots of different individuals or families with keratoconus, as many thousands of patients as you can get. You look at all the different genetic variants that you find with when you apply whole genome sequencing. Then you have a diagnostic test that is a commercial entity where you just go back in and resequence all those particular genes that you found associated with that disease. So you can go in at 5,000 times the targeted sequence resolution, and you're much more successful in finding a defined subset of disease-causing mutations. In this particular case, we can combine the TGF-beta-I, the entire gene can be sequenced, and we're able to find any novel genes. As I said earlier, often there's a mutation that we don't know exists. So if we get a unique or novel mutation in TGF-beta-I gene, this particular targeted resequencing would find it because it sequences the entire gene. There's also about 75 keratoconus related genes looked at in this assay, which looks at over 2,000 different genetic variants. And this gives what's known as a polygenic risk score for, in this case, keratoconus. This could be applied to any disease. There is one caveat that I would like to say, that you will all be aware that with keratoconus, just like with age-related macular degeneration here, retinal diseases, longer studies, much more known about the genetics of them. There are some genes that if you have a mutation in it, it has a low impact on your likelihood to get the disease and your risk. There are other genes that have a higher impact. There's lots of unknowns. There's also other comorbidities, in this case, hypertension, and lots of other environmental factors, aging, diet, or smoking. So I don't want you to go away thinking genetic diagnosis sits on its own. And again, that's the case with keratoconus. Very much this kind of multifactorial, it's a complex disease. There's lots of other characteristics and things that influence the disease progression and your risk of it, be that eye rubbing or other things. And obviously this genetic test would sit alongside all those advanced imaging methods that you all have. There's a glaucoma polygenic risk score as well that's now approaching the clinic, which would enable risk stratification. Again, that would facilitate a very personalized approach to glaucoma patients, where if they're very high risk, there might be earlier treatment options instigated versus lower risk patients may just be monitored. If you were to look for next generation sequencing panels and their use in ophthalmology, they're everywhere. They're in the NHS, they're in universities, they're in private companies, and there's a plethora of eye diseases that you can assess in terms of what genetics is playing a role there, and they're available as diagnostics. The turnaround time for these, I think, is relatively long at 90 days, and there is a cost there, as you can see, that it's a fairly extensive cost. So you would imagine that if you were going to be instigating these types of genetic eye disease tests, you would want to hold them for a reason. The patient and the clinician would want to be using them to dictate the clinical treatment or the pathway. And John mentioned earlier the use of Luxturna, and it's adding a copy of RP65 when there's 2 faulty copies of the gene. So it's very much you're doing this genetic diagnostics to inform your particular treatment, and in this case it would be a gene therapy when it is Luxturna. So that leads me on to gene therapy, and John has explained that gene therapy for a genetic disease can involve removing, repairing, replacing, or introducing a new gene where there's either a loss or a gain of function due to a mistake in the DNA. It's been on the go for a long time. Initially, there were some safety concerns and then some efficacy issues. I would say the efficacy issues, in my part, I think are down to delivery, and we have struggled as scientists globally to deliver these gene modalities. There's also over 2,500 clinical studies underway, and it's across a variety of diseases, and 6 gene therapies have been approved so far by FDA or EMA. I particularly do gene therapy and develop gene therapy in the cornea. Why the cornea? I think it's a monogenetic disease in terms of corneal dystrophy. There's a very clear genotype-phenotype relationship. It's quite well characterized. I think I naively thought the cornea would be easily to access, easily to deliver to. It would be a small surface area to treat, wouldn't have to go systemically with any of these molecular drugs. I assumed the disease status and the treatment success would be very easily observed and monitored with a slit lamp, and there's a relatively large prevalence allowing for clinical trials. And for something like Meesman's corneal dystrophy, the corneal surface and that epithelium turning over would allow for a complete recovery. So the cornea for me was the ideal place to start with gene silencing or gene editing. John mentioned there's one bad copy in some of these autosomal dominant diseases like corneal dystrophies, and what we want to do is try to attempt to look at the error where it is in the DNA that translates into the message. We specialize in developing what's called mutant or the bad copy specific siRNAs. These molecular scissors are designed to cut that constant supply of messenger RNA, stop the bad protein, and allow healthy cells to then grow and resurface the ocular surface. We also work on CRISPR-Cas9 gene editing, which is a once-off treatment. You only apply it once. The siRNA has to be a continual treatment every day because it is continually targeting a product of the DNA. And the CRISPR-Cas9 does a similar thing. It alters the DNA and for good, all in at once. I'll not go into the detail of any of this other than to say we do a lot of work on gene walks and developing the exactly correct type of molecular scissors that only cut the bad copy. They cut it with a very high potency, so we're able to have therapeutic doses in the nanomolar range. We are certain at every level it doesn't cut the wild type copy, which is needed for normal homeostasis and wound healing of the cornea. We assess it in multiple models in the lab, in corneal epithelial stem cells, and we have developed animal models. We've developed this animal model where we tag the bad copy and make it glow red. So the more of the bad copy there is, the redder the eye is in the mouse. As you can see there, the image is very red when there's the bad copy of the corneal dystrophy gene expressed. Due to the issues with delivery and delivering these into the eye, we do intrastromal injection, and you can imagine that's not quite so easy in the tiny little mouse eye. But here you can see we've treated the right eye, this eye on the right hand side, and we're able to reduce significantly by adding siRNA the amount of bad gene. Delivery is a problem. We have paired up with CySAF, an injection-free silicon drug delivery technology. This is a Belfast spin-out company now based in London. We've looked at topical application, so that is just applying this in the form of artificial tears. We combine the siRNA with this particle, the silicon particle, and we add it here to the right eye, and we do a live animal imaging. You can see it is retained in the right eye right up for 24 hours after one application. And the cross section here shows lots of this red glowing labeled siRNA penetrating through the corneal epithelium, through the stroma and to the endothelium, lending itself to deliver siRNA as a topical application for the different corneal dystrophies, be it Fuchs or be it the surface corneal dystrophies, Meesman's or Avellino or the other lattice dystrophies. Here we followed the live animal imaging of the amount of the gene expressed when we applied the drug daily for 7 days, and you can see we saw a reduction in all the animals, which increased when we did the washout period and stopped applying the drug. So it really is working. We looked at irritation, inflammation, and we're now at the stage where we're doing formal toxicology studies and biodistribution studies to ensure efficacy to move this through FDA for approval for a clinical trial. We've published this work, and it's definitely one of the first to show topical application of one of these gene therapies for the cornea, and we hope that this will progress in the future year. SciSaf and Avellino are actively working together collaboratively to push this forward to the clinic and raising finances to fund the next stage of the research. I would envisage this as something that would form a partner diagnostics and therapeutics, where patients will present with the disease or present for laser eye surgery and discover they have an unknown TGF-beta mutation. We are able to then, at the same time as doing the diagnostics, we are able to look at the actual personalized treatment that would be specific to them, to their genome, to their sequence of their DNA. And that would therefore be their own personal eye drop that would contain guides specific for them for a topical application to treat the particular corneal dystrophy they have. There are lots of other anterior chamber gene therapies in experimental phases, everything from glaucoma to scarring to wound healing, graft survival, keratitis and glaucoma, and I'm sure you'll be aware of the Editas, the CRISPR gene editing for Leber's is also underway at early stages of a clinical trial. There's a phase 3 late stage clinical evaluation for dry eye disease as well, a topical application of an siRNA. And with that, I would just like to say thank you, and hopefully that was very much a whistle-stop tour of gene therapies and genetic diagnostics. Thank you, John.
Thank you, Tara. Absolutely amazing work. And I have to emphasize, I've known Tara for a good few years, and she is one of the most diligent, hard-working research workers that I have ever met. And that shows in the work here and the amazing amount of work that's been done, not only identifying changes, but then working diligently to make sure they are the correct changes and the ones that need to be modified and modulated, and then to work on the whole mechanistics of how to introduce a corrective therapeutic system, and not to get bogged down with the bureaucracy of regulatory affairs. So it's ongoing and absolutely fantastic work. So I'd now like to hand over to another dear friend who I hope will have got some understanding of all the work that goes into producing a therapeutic component before the clinician can actually use it in the clinic. Paulo Stanga is someone that anybody in retina knows. He's a ubiquitous clinician, dabbles in lots of different areas, from artificial intelligence through vitreoretinal surgeon to photolysis, lasers, and gene therapy. So Paulo, tell us how all the wonderful work that Tara has done is actually ending up not only in the clinic, but in patient eyes.
Thank you very much, John, for this invitation and that kind of introduction. It's going to be difficult to do justice to the fantastic work that Tara is doing, but I'll try. So let me explain. You've asked me to explain as in a most simple way, a simple possible way, what gene therapy is and my involvement in gene therapy. I've now been involved in gene therapy for approximately 6 years. So in a nutshell, what is gene therapy? What is a gene? A gene is a segment of DNA And these segments of DNA are in our chromosomes, which, as we know, are in our cells. So genes are made of DNA, which produce proteins and enzymes. It is estimated that we have 20 to 25,000 genes. The DNA is composed of different nucleotide bases, and mutations in DNA composition lead to changes in genetic expression, and therefore changes in the expression and production of proteins. So again, what is gene therapy? It is the introduction of normal genes into cells in place of missing or defective ones, trying to correct genetic disorders. The idea is that this allows doctors to treat a disorder by altering our genetic makeup, rather than using drugs or surgery. We are trying to introduce a normal copy of a gene so that we can restore the function of a given protein. We are trying to treat diseases at the source, at the genetic level. We have to remember that inserting a gene directly into a cell usually does not work. So we need to use what we call a vector. And vectors are usually modified viruses. So we have to modify viruses so that they do not cause disease in people. And the two types of viruses that we use, either retroviruses or adenoviruses. My involvement has been using only so far adenoviruses. So the characteristic of adenoviruses is that they introduce the DNA into the nucleus of the cell, but the DNA is not integrated into the chromosome. And vectors can be injected either intravenously or directly to the target tissue. Tara made reference to how easy it is to target the cornea. Well, I'll show you how we target the retina. So what we are trying to achieve is, by delivering the vector, is to make a functioning protein again. So we want to modify genetic expression to obtain a therapeutic effect in either cancer or genetic diseases or infectious diseases. We are trying to replace a faulty gene with a healthy copy. We are trying to either inactivate a non-functioning gene or obliterate part of its function, or we are trying to introduce a new gene or a modified gene. We have two main methods to achieve this, either by using a patient-derived gene therapy or gene editing technology, which is what we are mostly using. So for this, as I mentioned before, we are using a vector, either viral or bacterial. I am involved with viral vectors. We try to inject the vectors directly into the targeted tissue. We are trying to diminish the potential for side effects. Now, this poses challenges, of course. The main challenge is being able to target the tissue, to reach the tissue. For example, the retina, which is, as we know, internal to the surface of the eye. We need to be able to successfully deliver the genes and, most importantly, avoid an immune response. There's a lot of talk at the moment about mutation-specific versus agnostic therapy. Most of what we are doing at the moment is mutation-specific therapy, at least in retina. And this is where it's becoming very difficult, because if we look at, for example, retinitis pigmentosa, which is one of the conditions that John mentioned at the beginning of the webinar, there are over 120 mutations that are responsible for RP, retinitis pigmentosa. It's going to be very difficult for industry to keep up and have 120 different gene therapies for the different types of RP. So we are looking more and more into agnostic therapy, which means using the same approach for the different mutations. And here I would like to draw your attention to Nanoscope Therapeutics, optogenetic gene therapy, optogenetics is really gaining strength, momentum at the moment. And for example, this is injecting opsins, which are ambient light sensitive molecules intravitreally. And the idea is to confuse the remaining cells, viable cells, into believing that they are photoreceptors and capturing light. So this is a very promising area. So let me start with my experience in gene therapy. This started in approximately 6 years ago. This is the paper we published in Nature Medicine using gene therapy for X-linked retinitis pigmentosa. So here I'm showing you the surgery. This is myself operating. This is intraoperative OCT. Here you can see a thick partially detached internal limiting membrane. We do a full vitrectomy. It is important to remove as much vitreous as possible, so there is no chance of remaining vitreous trapping any vector. You can see that I'm peeling internal limiting membrane, how the peel pulls the retina up. This is very, very thin retina, atrophic retina. And you don't see this unless you have intraoperative OCT. I'm creating a first bleb, a subretinal bleb. You can see how the tissue, the neural retina is elevating, and it is within that bleb that we inject the vector. So it's a two-step approach, a first bleb and the injection of vector carrying the gene therapy in the bleb. This is a bubble of air at the end of the surgery under the retina. We presented this at the 2019 American Academy of Ophthalmology, and we received the Best Paper Award. I presented for the whole team. This is another example of the surgery, the 2 steps. Here we are preparing the sample in theater. And as you will see, we create the bleb, and it is very challenging in inherited dystrophies because the retina is very thin, as you know. And here again, we see the bleb, you see the fovea there, very thin, and it's important to make sure that the fovea does not rupture. So there are other modes of delivery. So there are several modes of delivery. Intravitreal injection is also gaining momentum because it is easier. The patient does not have to go through a vitrectomy surgery. This is a diagram that shows the different types of approaches. Intravitreal injection, by far, of course, the easiest. Subretinal injection, which is either transvitreal or transchoroidal, and suprachoroidal injection. So 4 modes of gene therapy delivery for retinal diseases. These are some of the studies I am currently involved with in gene therapy. The ones in red have completed recruitment. SCOPE was and it's an ongoing study, but recruitment is closed, is an observational study. FOCUS is a phase 1 and phase 2 study. This is gene therapy. It's a study sponsored by Gyroscope. The company sponsoring is called Gyroscope. The product is GD005, and the aim is to modulate the complement system. As you know, it is now believed that a proportion of patients with geographic atrophy in late dry AMD have an unbalanced complement system. As you know, the complement system is part of our immune system, and this leads to either, for example, with this study, we are trying to increase the production of complement factor I, which is reduced in a proportion of patients with geographic atrophy. So let me show you some of the inclusion criteria, patients obviously over the age of 18, dry AMD with geographic atrophy in both eyes, and of course, no history of choroidal vascularization or any other intraocular treatment. These are some of the fundus autofluorescence images of eligible patients. This is for you to see the size of lesions that we can include in the study. As I'm sure you know, autofluorescence clearly shows the areas of geographic atrophy with loss of hyperfluorescence, what appears dark. What appears hyper-autofluorescence is the transition zone, and these are the areas of RPE that are under stress and are the areas that will undergo geographic atrophy in the near future. These are non-eligible patients. These lesions are too large. Usually, the cutoff size for lesions is 17.5 square millimeters in most studies. So what we are trying to do is trying to make the eye produce the treatment with a one-off surgery. We know from studies done by Panelists that if we can modulate in a positive way by blocking the cascade, we can slow down the progression of geographic atrophy by 30 to 50% per year. Hence the idea of gene therapy to make the eye produce the drug without the need of monthly or bi-monthly injections. So this was the first surgery that I did for geographic atrophy. I am the London surgeon for the gyroscope study. As you can see, a very large team of people supporting me. And again, it is a two-step approach. The technique is very similar to what I have just shown you. This is an example of the area of geographic atrophy. And let me share with you, this is a presentation from the last Arvo. This was delivered by Dr. Nielsen from the Wolf Eye Clinic. And here we are all the different, this is myself here, we are all the different investigators that participated. So what we are trying to do is trying to amplify the production of a complement factor I by a one-off surgery rather than monthly injections. And we are seeing by obtaining vitreous cavity samples, aqueous samples, that there is an upregulation of factor I. And again, here you can see how the elevation in aqueous mirrors that of the vitreous. So the take-home message from this study is that factor I is a natural regulator of the complement system. And that GT005, which is the drug that we are, the investigational product, the IP that we're using, is well tolerated in patients with geographic atrophy. And this leads to significant sustained increases in vitreous cavity factor I after one single surgery. So what we can say at the moment to patients, and this is an ongoing study, is that we are achieving what we were aiming to do, which is making the eye produce the treatment. What we don't know yet, and this is being looked at, of course, is whether this is having the desired effect on the area of geographic atrophy. So gene therapy has significantly expanded, increased, grown, improved since the first gene of a genetic disease was isolated, the rhodopsin gene, in January 1984. We can now identify the gene in slightly over 65% of retinal diseases. John spoke at the beginning of the webinar on Luxturna and targeting RPE65. So how do I envisage gene therapy in the future? Well, vitrectomy, suprachoroidal surgery, even intravitreal injections. We are pushing more and more for earlier surgical indication, while there is still good vision. Of course, the initial trials were done in patients with advanced disease. Day case surgery, we are already there. Local anesthesia, with or without intravitreal sedation, we are already there. Short operating times, less than 1 hour, we are already there. I am currently doing the surgeries in 35 to 40 minutes. This is as part of studies, of course, where we are checking and rechecking everything. So it could be potentially 30 minutes procedure. Fast patient recovery, we no longer use systemic steroids. I'm sure you remember the concepts of Cirora and Irora. Cirora, complete RPE and outer retinal atrophy, and Irora, incomplete RPE and outer retinal atrophy. We really want to start treating at the stage of Irora, not Cirora. And then we have a chance of turning back the clock, perhaps, as there's been some work presented by Michael Ip at the Macular Society last year with the Apellis drug. So thank you very much. I would like to thank my colleagues, my team, Dr. Ursula Reinstein, Ruth Goddard, Nathma Faruqi Bashir, and Julia Reinstein. And do not worry, please, because we will get your AMDs. Thank you very much.
Thank you, Paulo. I know you're going to rush off, so let me get 2 elements in quickly before we ask Tara some questions. Complement. Are you sure that complement is a cause, not an effect? Because complement changes only seem to occur after loss of outer blood retinal barrier. That's the first question. The second question is, why have so many retinal gene therapy companies not gone bust, but lost a lot of money?
Well, those are 2 very important, very good questions, and very good questions are difficult to answer, but I will try.
Sorry.
I wasn't expecting anything less from you. The first question, is it a cause or not? Well, I don't think we really know. What we do seem to know, as I've shown from the work that's been done, is that if we increase, the gene therapy is able to increase the production of complement factor I, but most importantly, perhaps, what we do know is that if you manage to regulate the complement system by blocking either C3 or C5, so C3 at the beginning of the cascade, C5 later down the cascade path, you can slow down the progression of geographic atrophy. There are 2 companies that are looking and have been looking to this, Apellis and Iveric Bio. Apellis targeting C3, Iveric Bio targeting C5. I've got a commercial association with Apellis because I am an investigator in the Apellis study, and I was the London investigator for Iveric Bio, but then when COVID came, that was stopped. So we are hoping to be able to restart the Iveric Bio research in the UK very soon. And Apellis, both have obtained very similar results targeting C3 and C5. Let's concentrate on Apellis. Apellis last week filed an NDA with FDA, a new drug application. That confident is Apellis of the results of the studies. And this goes back to to your second question, why did so many companies have gone bust? Well, I think the most obvious answer is because they have not managed to succeed, showing that there's been a successful response. And as you know, all these companies rely on investors, and investors want to see their money. back, understandably. So if you cannot show results, investors pull their money away. So Apellis has shown results. In fact, there are publications already in the public domain, and with every monthly injections, you get almost 50% of slowing down the progression of the disease. With monthly injections, close to 30% of the progression. So ideally, you would like monthly injections. Of course, that may be difficult. So we'll see where we end up with the best possible regime. We've now completed the phase 2 study, the OAK study, and now we are embarking on a phase 3 study where every patient is receiving the treatment. The phase 2 study, the OAK study, and identical studies carried out in the US. They were randomized to, patients were randomized to either placebo or treatment, and to either monthly or every other month injections. So it seems we're going to be having, an approved treatment for to slow down the progression by next year.
Yeah, my concern is always that a lot of companies make a lot of money out of treating end-stage complications and not the fundamental etiology of the disease. And we've seen that in relation to the whole herd of anti-VEGF treatments. Yeah, sure, you may control the new vessels for a bit, but you're not actually controlling the disease process.
It would be ideal, of course, to target the disease earlier, either with 2RT, laser, or other technologies to try to turn back the tide much earlier. And as I mentioned before, ideally, we need to start treating at the stage of iRORA. That's why we are really, I think we are really going to change the way we look at AMD, dry AMD. I'm sure you remember everything we have to go through to convince people that with wet AMD that was treatable. We are now at the stage when people with dry AMD have been discharged. They are no longer in the system because they've been seen and they've been told nothing can be done, go home. I still see patients, we're still screening for patients, and they've been told that, yeah, there's no treatment. So we are working very closely with, for example, with the Macular Society to try to bring the patients for screening. I think screening is going to be fundamental. We're going to move to screening of intermediate AMD. That's, I think, the future, because that's where we need to start treating intermediate AMD.
Now, but Paulo, you've actually given a lovely segue for Tara. Screening is important. Tara, screening, genetic diagnosis for screening.
Absolutely, John. When it's a family and not a sporadic gene causing disease, of course, with the families, you're more aware of it. The parents are more aware that possibly half their children might have it, so they're acutely asking for screening. But in the sporadic disease, the sooner and the better the screening is, and the broader ranging to catch whatever is causing the symptoms and treating that before damage is done. As you know, once some of the damage is done, and Paulo has already mentioned, he would rather be starting the therapy much earlier. So absolutely, the more widespread and the more take-up there is of genetic screening, obviously the better it is for any of the genetic diseases, including the eye ones.
Okay, and just one comment, if you would. The prevalence of keratoconus is pretty variable depending on the study you read. How important do you think, and how accurate do you think, is the Dutch study with approximately 1 in 400? That is out of step with all the other studies.
But that may be a reflection of more genetic testing, the uptake of genetics, and in many countries it's just not looked at. People aren't testing, aren't looking for keratoconus, and they aren't considering the genetic influence of it. So it's probably relatively accurate, but is not reflective of other countries because they're not being tested, they're not being assessed for their keratoconus polygenic risk genetic factors.
But it's probably higher than we think.
Well, yeah.
Chris, you had a comment.
Yeah, just on the point about screening with especially with intermediate AMD, screening misses a lot because you have to have some form of damage present because most of the screening is done with say OCT. So can genetic testing play a role in the screening and identification of intermediate AMD much earlier than what we're seeing today? And similar to that point with keratoconus, is it more is it going to be key to the success of genetic testing that it be used as screening rather than as a sporadic thing that clinicians employ when they suspect something?
Yeah, very much so, Chris. I mean, that's the point with corneal dystrophy and the screening for corneal dystrophy. The DNA mistake is there before the symptoms. The DNA mistake and those SNPs and those mutations that we look for are already there, but the patient won't even be aware of it. They have no symptoms, they have no pathology, and it's not until there's an injury or they undertake elective spectacle-free surgery that they then end up accelerating the disease and the phenotype and the symptoms. So those diseases and that kind of perfect storm of environmental factors and genetics and everything that goes along with a disease progression, they're there from the start. So if screening was cheaper and more accessible, of course, we would be discovering things much earlier. It then becomes whether or not we are a culture for using gene therapy. Will we be a culture where we will adopt and be very easygoing about, yeah, I'll have that gene therapy and I'll have that gene therapy. It's almost as if we wait until it's the worst case scenario in terms of the disease symptoms and then go for gene therapy.
And Tara, if you take something like AMD, the genetic defect is there from conception, but the manifestation isn't until the 6th, 7th, 8th decade. So do you think there are, as it were, clock genes which govern the onset time of a defective gene in an age-related disease?
I think there are so many complexities in the human genome, and it's a real milieu of epigenomics and methylation, and so many things go on within the cells that we probably don't know enough about it, but for certain that can be accelerated. And you can see that in some families with an R124C mutation, their corneal dystrophy can be really aggressive and be quicker. So there are other complex factors going on within the genome when it's genetic, as well as all the external other factors such as environment and comorbidities.
Michael, you had a question.
Yeah, I spent some time in my life on keratoconus, and it's just more of a practical question. So let's say I do request someone doing a genetic test. There's an indicator that you might have a risk for keratoconus. So what's next? I mean, what's next for the patient? So what's the consequence here? Are we still doing screening with topography, or are we doing an earlier intervention with cross-linking? So my question, I think, is basically saying, okay, great, I know I have a risk factor in my genomics for keratoconus, but does it change anything in the course of our treatments, or we just don't know yet?
Michael, before passing that on to Tara, for almost 50 years, I've been associated with the patient group for inherited retinal dystrophies. And it's very interesting, the culture within that group, because there are a number of individuals, a minority, I have to say, who absolutely want their children tested for a genetic condition. The vast majority say, until you guys have a treatment, I'm not sure I want to know. And that's an interesting cultural element here. And clearly, the closer we come to treatment regimes, the more important this screening becomes. And in something like retinitis pigmentosa, a very wise clinician said to me, it's a dustbin diagnosis, and it's rather like describing a man with a limp. Now, you can limp because you've broken your ankle, your shoes are too tight or you have a stone in your shoe. It's no good giving new shoes to a guy with a broken ankle. So that's why it's very important to subset the genetic types, because when treatments come along, we already know and you can be put in that group. Tara, would you like to comment?
Yeah, I agree with everything that John has said, and it is a very valid question, Michael. It's almost like me saying, what's the point in having a genetic diagnostics if you don't have a treatment? In time, there may well be some gene therapy treatments for keratoconus, but at the minute, it's so polygenetic, so multifactorial, that's not an avenue anyone's going down. I mentioned the polygenic risk score. If that score was very high and your genetics and your makeup was telling you you're at really, really high risk, and if I was to tell you you've got gene mutation A, B, and C, and that gives you a very high risk score, I'm sure you would monitor more closely and you would consider an earlier instigation of some of the treatments. But again, it's all to do with everything else coming together. Are you an eye rubber? Are there other factors that are going to accelerate that disease progression? What age are you? What are you hoping to do in terms of career? You know yourself everything about the management of keratoconus.
And following up to Michael's comment, it'll be very interesting, Tara, I think, downstream to see if there's any cross-correlation between a genetic diagnosis of KC or risk factor and the response of individual genetic groups to cross-linking.
Yeah, I can't answer that. It would be interesting to see. Yeah.
You stole my question, John, but that's where I was getting to. But exactly. So now we have kind of understood there's some sort of a genetic pre-programmed situation. Are we getting different responses from certain therapies? I mean, very good point.
Yeah, I think that's very important.
John, we've got somebody who wants to ask a question. I'm going to let them talk.
Yeah.
Tina?
Hi, yes, hi. Thank you very much for the wonderful presentation. Lovely seeing you. Having you mentioned about the autosomal dominant RP, and now we have gene editing, and also we could knock out, dual knockout and knock in. One of the key barrier and the challenge, I think, is you are able to completely silence the mutated protein, the genes, and in the protein production process stage. But the level of the protein expression, the knock-in part, will only achieve 1/3 in both the gene editing data by Adidas and Everett Bayer, which was the work done by UPenn. They only can get 1/3. I think my question comes to the point is, if we really need to make the gene therapy to become really take a leap, not just moving from hand motion, navigating things to be able to just a little better. You need to help people to live a normal life. In particular for RP, you can have early diagnosis, early treatment. If you are only can address 1/3 of the protein production, is it enough? And the second question is around the gene editing efficiency. The moment look at the Adidas program, they seem to have not shown significant problems of safety, causing, cutting the bad area, run cut and whatever will cause a mutation of oncogene insertion like lentivirus had, in some of the clinical indications. But anyway, the key challenge, I think, is that at efficacy, according to NIH, NEI's the paper, they advise Adidas that 10% the efficiency at the population level, not the individual level, seems to be enough. And that's not enough. If you can only correct the 10% of photoreceptor, that is not enough to generate it. It's not good enough, sufficient enough to generate enough photon to produce meaningful vision acuity. In the center region, maybe with light perception or sensitivity improvement, that certainly is not enough. So these are the two technical challenges. I wonder how John would take it. I have several others to Tara and Paul. The Tara was for the delivery. I the 80% will get like my dog, can you use the bandage for 24 hours and reduce the systematic exposure? That's another one. And so I leave it here.
Well, Tina, I think that I'm mindful of the clock because we have to keep to time here. And I'll just let Tara, is there anything you wanted to pick up there?
Tina, I agree with everything. To be brief and as quickly as possible in answering, delivery has always been an issue. Autosomal dominant, we can get off with reducing just half of the silencing, of the bad gene. But for replacement, yeah, we do have to replace more. And anyone working in delivery and delivering these genetic therapies, that is the area we need to be looking at. The bandages or the lens, as you suggested, I answered on the text there. We can't injure anything, or we have to be careful of any kind of trauma when we're dealing with TGF-beta-I, because we would just induce more disease with injury. But we're looking at slow release as a way forward rather than daily application. But yeah, very nice comment.
And I think, Tara, your dosing in treating autosomal recessive disease with terms of gene augmentation, that is a problem. And I think that is the reason why so many of the early companies lost money and have decided to close. Well, I'm going to close this meeting because I think we've had a very good discussion. I hope it's been informative. I hope it's helped those that haven't got a primary interest in genetic therapy. I hope this is tempting and gets you involved. And then I want to tell everybody that the next Oracle is going to be on August the 9th. I do not know its subject matter, but I'm sure it'll be interesting and exciting. And thank you all for coming here this evening. Thank you.
Thank you, John. Thank you, Tara.